Hi and Assalamualaikum. I'm Natasha Danya and I'm from D386. So, today we will discuss about the Biology Mastery A6 that can help you during your final on June. Before that, I would like to apologize if I don't look straight at the camera because I'm looking at the slides. Alright? You okay with that? Kalau you nak I tengok, you pun boleh. Tak adalah. Okay, now, let's start. For question 1, why most ecosystem can only support 3 or 4 trophic level? So during these slides, I will just give points and make sure you have these points in your essay, okay? So the points are energy losses between trophic levels, energy loss as heat or via respiration, excretion, metabolism or movement, and after the 5th trophic level, only small amount of energy is left. Energy is insufficient to support the organisms in the 5th trophic level. You get that? Now let's move on to question 2a. It wants you to compare continuous and discontinuous variation. So for this slide, I have separated it. The left side is the similarities, the right side is the differences. So as for similarities, it caused by genetic factors and it may occur during meiosis. While for differences, now we look at continuous variation. It is quantitative inheritance such as height, weight, and skin color, anything that can be measured, and it has no intermediate phenotypes. It controlled by polygenes, which mean many genes affected by genetic and environment, and it has normal distribute curve, or we can call it as bell-shaped curve. While for discontinuous variation, it is qualitative inheritance, anything that cannot be measured. Take note. Such as ABO groups, ability of tongue rolling, ear lobes. It has no intermediate phenotypes, controlled by single gene, affected by genetic and environment, and it has discrete distribute curve. You get that? Okay, this session kita relax je, tak payah attention. Okay, you can take a pause if you want to drink or you want to rest for a bit. Yeah. Now we look at question 2b. It wants you to describe the factors that may cause genetic variation. Before that, genetic variation has 2 types: genetic sources and environmental sources. We will look at the genetic sources first. It has 3: mutation, chromosomal mutation, and crossing over during prophase 1. In mutation, changes in genetic material could occur spontaneously. Now, look at the key point: spontaneously. Gene point or point mutation refers to changes in the base sequences in segment of DNA molecule. While in chromosome mutation, it occurs when changes in chromosomes number or the chromosome structure. During gamete formation in meiosis, recombination of genetic material results in gametes with different genotypes being produced. In crossing over during prophase 1, it leads to their genetic material being exchanged. The fusion of male and female gametes during fertilization also brings about recombination of genetic materials, producing zygotes with different genotypes. Okay, that's for genetic sources. Now we will look at the environmental sources. It only involves phenotype differences and is less important in evolution. Usually this version is the result of interaction between the environmental conditions and genetic sources. Oh, I misspelled the environmental. It's fine. The environment is capable of causing changes in gene frequency and genotype frequency in a population, but it doesn't change the genotype of individuals. Yeah. Now, let's move on to question 3. Explain the mechanism of enzyme action based on induced fate model. So, the points are: bring substrate closer to each other, facilitate the breaking of bond of conformation between substrate. A substrate binds to an active site, conformation change shape slightly, and when an enzyme binds its substrate, it forms an enzyme substrate complex. Easy kan? Now look at question 4a. Explain the NADH production in Krebs cycle. So first of all, isocitrate is oxidized to become alpha ketoglutarate. Alpha ketoglutarate. Susah betul aku nak sebut. And AD is reduced to become NADH, and AD plus is reduced to NADH plus H plus. Again, alpha ketoglutarate undergoes oxidation forming succinyl CoA, and AD plus is reduced to NADH plus H plus. Besides that, in oxidation, malate is oxidized to oxaloacetate by transferring 2 hydrogen atoms to NAD. NAD plus is reduced to NADH plus H plus. And now, let's quickly move to question 4b. Describe what happened to FADH2 that is produced in Krebs cycle. So points are oxidation intercellular respiration halt the electron transport change. FADH2 oxidized to FAD, dihilang hydrogen. That's why it becomes FAD. Since FADH2 has less energy than NADH, coenzyme Q oxidoreductate. Susah lagi nak sebut. It can only pass the electron to succinate COQ oxidoreductate complex to succinate dehydrogenases will undergo reduction. Tips for you guys, you need to always sebut the words, the complicated words, so next time it's easy for you to pronounce it. Now, as you can see, it's hard for me to pronounce it, right? Now let's move on to the question 5. Explain Calvin cycle. As you guys know, Calvin cycle has 3 phases. Phase 1 is carbon fixation, phase 2 is reduction, and phase 3 is regeneration of RUBP. We will look at phase 1 first, carbon fixation. RUBP combined with 1 molecule of CO2 to form unstable 6-carbon intermediate. The reaction is catalyzed by ribulose by bisphosphate carboxylase rubisco, and the unstable 6-carbon will break down to form molecules of 3-phosphoglycerate. Now let's move on to phase 2, which is reduction. 3-phosphoglycerate is converted into glyceraldehyde 3-phosphate 3-GP using reduced NADP and ADP. NADPH then is oxidized to NADP+. The last phase is phase 3, which is regeneration of RUBP. 5 molecules of G3P are used in regeneration of 3 molecules RUBP. It requires ATP, and 1 molecule of 3GP is used to produce sugar. Okay, I think that's all. We will see each other again later. Not later. Just wait for my another video for question 6, and yeah, you will see later. Thank you for watching. Bye bye.